All right, time for chapter 5. So chapter 5 is our first kind of more interesting chapter. We're going to look at how cells work together to form tissue. So the histology word for our chapter title is really just a fancy way of saying the study of looking at tissues. So for our tissues, they fall into 4 primary tissue classes. We have epithelial tissue, connective tissue, nervous tissue, and muscular tissue. So for your tissues exam, which is your first lab exam, part of what you'll be doing is identifying the pictures, whether or not they fall into the epithelial, connective, nervous, or muscular tissue categories. And then there are other things that you'll have to identify, such as certain features or the specific subclass that fall in these 4. With these different tissue types, you have some organs that are made up of multiple types of these types of tissue. So for example, your brain is all nervous tissue, or your skeletal muscle system is all muscular tissue. But then you have things like your stomach, which is epithelial tissue on the inside, muscular tissue in the middle, and then connective tissue on the outside. So with our 4 tissue classes, they differ from each other in the types and functions of their cells. So for example, your muscular and nervous tissue, they both can be stimulated. Your epithelial tissue, it's usually facing some kind of outside environment. They're usually closely packed together, versus your connective tissue, they're a little bit more spaced out. The matrix between them is different. So epithelial, pretty much very little space in between the cells. Connective actually has actual extracellular material, just more than just fluid. So you might have different strands of proteins that are there to fill in the space and provide some structure. And then how much space is there versus the matrix? So again, epithelial, very closely together. Connective, many times are mostly spread apart. Your muscular is pretty much packed together. Nervous, within the structures of the nervous system, they're fairly packed with each other to an extent, but they also kind of spread out a little bit. So they all have different spacing, different ways of looking at how the tissue looks, and you will be able to see like different cell shapes, and that also helps to determine your different, not only primary class, but also the subclasses. Okay, so for my respiratory therapy students, I know you definitely need to know how a slide is prepared and understanding what you're looking at. So this is very important for you. For the nursing, I know you guys definitely have to know how to use a microscope, so paying attention in the lab is good, but this part is also good so you actually know what you're looking at when you're working with a microscope. Okay, so when we get our tissue slides, and we'll see this in lab, the people who are making the tissue slides, so our histologists, they're going to take a 3D object, so a section of tissue, they'll slice it really thin so that when you shine light through your slide, and so we have our little piece of tissue here, it's thin enough for your eye to actually see what's going on in the sample. And sometimes you actually also will stain it so you can see better on the structures. So depending on how the tissue is sliced is going to determine how much information you're going to get. So if I'm looking at this little egg, if I cut way out here, I'm only going to get so much information. So I'm totally missing this middle portion, where if you're looking at a cell, would be like your nucleus. So in this section, I would be missing the nucleus completely. Versus if I went a little bit further in, I get part of the information. I do know, okay, at least this middle portion is also partially on this outside portion of our cell, or in this case, our egg. A one slice down the middle gives me the full length of our specimen, so the length of the egg as well as the full height of our nucleus egg yolk, versus the ones out here, it's a lot shorter. So if you looked at the top of that one, you'd still have a little piece of information that you're missing. So if you're looking at, for example, a tubular structure, so our little elbow macaroni noodle, or something that is more similar to what's called like a sweat gland, is kind of like this, where depending on where you cut, then you end up having different forms of information. Sometimes if you're cut, for example, this way, you end up seeing only part of the information. So you don't actually realize that this whole thing is connected. So that's why sometimes you have to look at multiple samples in order to get a full picture. So here's another way, just different sections. Longitudinal, you'll get all of your lengthwise information, but you're not getting widthwise. So again, multiple samples needed. Sometimes you have what's called an oblique section, where it's done at an angle. And that angle is because maybe sometimes, for example, the substantia nigra, which is in your brainstem, it's at an angle. So every time that is shown, the cut is at an angle so that you could see the full structure of the substantia nigra. Okay, last two ways of putting stuff on a slide. So not always do you have something that's a solid thing that could be sliced. Sometimes it's more of a liquid situation, for example, blood, which is smeared or rubbed across the slide. The other type is where your tissue sample is very loose and doesn't really have a solid structure, where the sample is just kind of spread out on the slide. So, for example, this one here, the areolar tissue, so our little dark spots are the actual cells. The spaces in between with all the lines are just fibrous proteins. So that's not going to be too solid of a structure, and so you just kind of smash it down and make it really thin. Okay, so first major category of our tissues is epithelial tissues. So our epithelial tissues are found in places where they're exposed to the outside environment. So our skin would be epithelial tissue. For example, our digestive or reproductive or respiratory systems, because they're still exposed to the outside environment, even though they're internal in our body, they are still lined with epithelial tissue. So the jobs of epithelial tissue is to protect deeper tissue. So they're going, because they are the ones that are exposed to the outside environment. They can make different secretions. So for example, sweat, excrete waste out of the body. They can absorb different chemicals, including nutrients. So, for example, the epithelial tissue in your small intestines. They can filter stuff out, so kind of guard against what coming in and out of our body, and then also send stimuli. So, for example, like your epithelial tissue of your skin, it has sensory receptors. So with our epithelial tissue, the cells are closely stuck together, so lined right up to each other. Tend to cover body surfaces like skin or line body cavity. So for example, the inside of our respiratory system. They do not have blood vessels. So in order for the epithelial tissue to receive nutrients, it depends on the blood vessels from the connective tissue that's below it. So all epithelial tissue has that connective tissue that's below it. Bunch of blood vessels here, and then the nutrients will diffuse going from high to low concentration towards your epithelial tissue. So with our epithelial tissue, just mention this as well, the upper surface is what's actually exposed to the outside environment, and then your basal surface, the one that is not on the opposite side, is what goes towards the inside of the body. Okay, so epithelial tissue. First thing, high rate of mitosis, because they are exposed to the outside environment, so they're going to be damaged more often. All of the cells align on what's called the basement membrane. So one way when you're identifying your tissues and you're looking and you can actually see that basement membrane line on your epithelial tissues. That's also what's going to separate the epithelial layer from the connective tissue that's below it. So your basal surface is on your basement membrane. Your apical surface is on the opposite side. So in our epithelial tissue, we have different subclasses. So the first one being simple epithelial. So simple because it only has one layer, and of course it's epithelial tissue. So in this case, all cells are going to align or touch on that basement membrane. Next category is stratified. So stratified epithelial has at least two layers. So there's at least one layer that's going to be stacked on top of the other. So our first layer lines on the basement membrane, and then there's at least one more layer that does not touch that basement membrane. So it's on top of our bottom layer of cells. This other one that is put in the middle right here, that's actually still simple epithelial, but it actually has its own category because it just looks quite different. So this one is called pseudostratified epithelial tissue, and the columnar is just because of the shape of the cell. So we'll get that in a second. So the pseudostratified is because it looks like the cells might be stratified, like they're stacked on top of each other. Like, for example, this cell looks like it might not touch the basement membrane, but even in this case, all of the cells touch your basement membrane. Another way to classify epithelial tissue is based off of shape. So down here we have three different cell shape types. So we have squamous, which is our flat fried egg, cuboidal, which looks like a cube, so it's going to be equally as wide as it is tall. And the last one is columnar, which is tall like a column, so taller than it is wide. So going into our different subcategories, we have simple epithelial. So we have our one layer cells in either the squamous, cuboidal, or columnar shape. And again, pseudostratified is still a simple type of epithelial. It looks like it's stratified, but actually every cell touches the basement membrane. So in our simple epithelial tissue, we do have a type of cell that does hang out in this type of tissue. So again, your epithelial tissue, usually it's facing the outside environment. And so a lot of times this also means that the tissue needs to be protected. So one way to protect that is by creating a surface coat of mucus. So for example, like in your respiratory system. So those cells are called goblet cells. So they get their name because they look like goblets. So here is a wine glass. Goblet is just an old way of saying that. So down here at the bottom, this is the cartoon version, but still works. So our little goblet-shaped cells, so their job is to make mucus. That's all they do. They make mucus, they excrete it out, and that helps to coat our tissues and protect them from damage. Okay, identifying our different tissues. So here we have, this side is the microscope side. This side is our cartoon version that kind of helps to point out stuff, makes it easier to see. So remembering your epithelial tissue is always partnered up with a connective tissue below it. So here are connective tissue with all the space. That's why you see all this other stuff here where it looks like, okay, if you're trying to identify stuff, you do have to pay attention to what also might be in your sample. So again, simple squamous epithelial tissue. So here's our basement membrane. One row of cells, so it's simple, and then squamous because it's that flat fried egg, sunny side up shape. Next one, simple cuboidal. So this sample is from your kidneys, and there are a lot of tube-like structures in your kidneys. So even though it looks like there's a bunch of different layers here, we're still looking at our basement membrane here, and then our cells are along that same line. Cuboidal is the shape, simple because it's only one layer of cells. Job of simple cuboidal, absorption and secretion. I'm going to go back to our simple squamous. So this one is for diffusion. So very thin gap space between our like the thin membrane between the cells. So that's the function of our simple squamous. Okay, so we've done our simple cuboidal. Simple columnar epithelial. So this is where we get our goblet cells here. So again, here's our basement membrane, column-shaped cells, and the goblet cells are just here. You actually can see their shape. So with our simple columnar epithelial, it has a brush border of what's called microvilli. So if you remember that from chapter 3, this is just cell extensions that make it better for absorption of different nutrients or different chemicals into the cells. So here in the microscope picture, you can't really see too much on the details, and that's why it kind of looks like a brush. Like everything is super packed because these tiny, closely spaced together microvilli. Okay, last one in our simple category for epithelial are our pseudostratified epithelium. Got some more goblet cells here. Even though this looks like multiple layers, it's not. They're actually touching the bottom of that basement membrane. So every single cell is going to touch. And in this one, instead of having microvilli, it has cilia. So remembering again from chapter 3, we have our cell extension structure, except for this one is going to help push that mucus along the surface. And for example, respiratory system, push it all the way up to your back of your throat and you swallow it. Normally the mucus is very thin, so you don't realize this is happening unless you're sick and then your mucus is really thick. Okay, moving on to stratified epithelial. So at least 2 layers of cell. It could be 20 or more layers. So for example, like on the bottom of your foot, really calloused, thick skin. Yeah, that's going to have way more layers of cell. Okay, so in this one, only the bottom or the deepest layer is going to attach to the basement membrane. Everyone else stacks on top. Same thing like our simple epithelial. It's based off of our shape of cells for our different subcategories. And then we have a fourth type, which is kind of cool. Okay, let's get our fourth type being the transitional epithelial. Okay, so stratified epithelial is our most widespread one, because it's skin. So in this case, at our bottom layer, that's where we get our new cells because that's where the nutrients are. So our new cells are going to push the older ones up. Eventually, the older ones are going to be so far away from nutrients that they'll die and flake off, which is also called exfoliation or desquamation. So in our stratified epithelial, we have 2 different types. For stratified squamous, they're keratinized, so this is like our skin, which is abrasion resistant, so this helps keep that tissue tough and less likely to suffer from like getting cut or scraped off. The other type is non-keratinized, so this doesn't have the dead skin layers because you don't have that extra toughness from the keratin protein that's been bundled up, and so those top layers of cells are more likely to be exfoliated off. Okay, first type, keratinized stratified squamous epithelial. So this is our keratinized one. So we have the keratin protein that gets bundled up, makes the upper layers very tough, very difficult to exfoliate off. So as our cells go from our basement membrane up towards our surface, they tend to flatten. That's where we get our squamous shape, stratified because it's multiple layers. And then here in the cartoon picture, which you can still see very easily in our regular picture, this is actually the layers of dead skin cells on the callus of your foot. So here is just that they're super tough, and that just keeps the tissues from being damaged. So just protective layer on our skin. It's just extra thick in certain areas. So sometimes I get this question, so I might as well answer it. So some people ask, what is this zigzaggy looking thing here? So for that one, that is and we'll talk about this in chapter 6, which is our skin chapter. In order to help keep our epithelial tissue and this connective tissue below it stuck together, we have these little interlocking folds in between the 2 layers, and that helps to keep them stuck together. Again, makes them just more tougher. So this right here is probably at the portion of skin where the connection's a lot deeper. So if I look at my hand, this connection here, that's most likely where that zigzag line is occurring. Touchscreen. Because of the deeper connection, so you don't have as much of a smooth area right there because you have a little bit of a wrinkle. It also helps prevent water loss from the skin, so keep us body from getting dehydrated, and keeps pathogens out unless you get a cut. Then that goes your immune system to activate. Okay, non-keratinized version. So here we have our basement membrane. Here's a connective tissue below it. Our bottom layer of cells, they're a little bit more rounded, but they flatten as they go up. This very top is where we have some dead skin cells that are going to eventually flake off. So example of this being inside the vagina. So it does going to resist abrasion just because there are so many layers, as well as prevent bacteria or some other pathogen from getting into deeper layers. Next one, stratified cuboidal. So stratified, more than one layer. So let's go to the cartoon because you can see it better. So our basement membrane, in this case being, this is a oil gland in the skin. So our basement membrane is this circle on the outside because it's a tube-like structure. And then we have our multiple layers of cells creating our stratified tissues. So sweat glands are like this. The ducts in the male reproductive system are also like this. As well as your glands that make your ovarian hormones in the ovaries. They're also this type of shape. Okay, last epithelial tissue, which is kind of cool, this one. So here's our basement membrane. And then this one's a little bit different. So instead of becoming smaller as you go through your layers, it actually becomes larger. So the cells become larger. And here's the reason why. For our transitional epithelial, it's in places where you need to have a little extra stretch. So in this case, in our duct system in the kidneys, as they fill with urine, you need that extra stretch, the extra stretch, in order to allow it to fill with urine. So if there's an influx that comes through, maybe you drank a bunch of water, maybe your blood pressure's up, you have space for that urine to be. Same thing through your ureters as you're going toward your bladder, as well as being in the bladder. All of it does is just provide stretch so that it's able to fill up. So you don't have to go to the bathroom every 5 minutes. You can fill up a little bit before you gotta go. Okay, we're gonna start the next video here.